here I have some organic chemistry in front of me. Oh, that's not the only thing that, yeah, all right. So there are other things besides organic chemistry, although it is very important. Yes?
Yes.
Thank you. At least one person agrees. Yeah. These guys absolutely agree. I know, because I've had all of them in my classes. And one of the ways that you can get to do a trip like this is to do research. See, this is generally senior level research, but it can also be work that is moving that direction or can result in a publication. Projects have been done with myself and with Dr. Kunzelman and with Dr. Baradovich that have resulted in publication of undergraduate students in the chemical literature, chemical scientific literature. So what the students are going to present today is their trip to the American Chemical Society meeting in San Diego, California. And as you all know, they had a very hard time over there. It was so difficult. They didn't want to be there. They wanted to be back in cold, yucky Michigan. But somehow they managed, as did the professors' wives, to have a really good time. The rest of the Dr. Kunzelman and I, we attended conferences. So that's enough. We have Kristin Rohr and Charles Shearer and Jen Kingsley and Kyle Thompson making this very hard-hitting, fast-paced presentation about their experiences in San Diego. It's going to cover chemistry and other stuff, some of it even fun. Take it away.
So, we thought we'd talk a little bit about our science first. Kind of tell you what kind of projects that we took. Here's some pictures. You know, Charles hard at work eating a bagel while presenting the poster. But we did poster sessions. So we had big posters that we tacked up on these bulletin boards, and then people would walk around, and if they found our poster interesting, they would stop and ask us questions. And the picture down here is the back side of the conference center?
Yeah.
The back side of the conference center that we were at. So I took my light stick research from Chem 112. This was my 3-week project that turned into a 3-month project, which turned into a publication in JChem Ed. So the background of what I was doing is when you break a light stick, a chemical reaction happens, and that releases energy. And that energy hits the dye molecules, it excites the electrons. They jump up to a higher wavelength, and then when they relax, they release a wavelength of light. And so I tried to figure out, well, what household dyes can I put into the light sticks to get different colors? Okay, so this one here is spinach in the light stick, and then here is the spectrum that I collected of what wavelength of light was coming off. This is red food dye, turmeric powder. This is a yellow dry erase marker, and a lead crayon pill that has quinine in it. And these are the distinct spectrums that were collected. And then I also did a set of dry erase markers, and they also changed. All of these started out as white. And then by adding a different dye, you get a different wavelength of light released. We tested that scientifically by comparing the spectrum of the white light stick, which is in gray here, with the spectrum collected after we added the dye.
I'm going to talk about the LEGO periodic table, which is just upstairs, outside the big lecture hall upstairs, and actually a lot of you were involved in. As most of you know, this was a part of our science outreach event, Halloween in the Science Lab, a way to take science to the community, and specifically to the children of the community, trying to make science fun and interactive and get them interested in science from a young age. A lot of you actually helped us with taking the little pieces of paper and putting the correct number of Legos that were indicated by that in a baggie with a little piece of paper. And then we hand them out to the kids at the Halloween and the Science Lab, and you actually see the kids. You actually see the kids building them here. They had a lot of fun doing it. It was actually really fun to walk around and see the kids putting the Legos together. The one issue with having children do it, though, is that they're wonderful and they're fun, but when you get the really little kids, they're not always going to follow the directions quite the best or build things they want to. So we ended up with some elements that didn't exist, and elements that used letters in the name that also didn't exist on the alphabet. So we had to do quite a bit of work trying to get the Lego periodic table to actually look like the periodic table and actually have some of the correct, all of the correct elements. And as you can see, we did is sitting upstairs outside the big lecture hall, and it's really cool. And a lot of people who are not science majors on campus also walk by it and see that it's cool and see that science can be fun. So it's getting to be a continuing outreach event, because not only is there seeing it, but there's also other stuff on the periodic table.
No, I didn't.
There's little Lego figurines that connect to the element that they're on. Like we have a guy holding a banana on potassium, or a little skeleton guy on calcium, just all these things that connect in such a way to continue to use it for education.
Chemistry is so much fun.
So do you have a miniature Dr. Baldwin for carbon?
We're looking.
I talked about mini GC analysis of a green combinatorial transesterification, which is a big long thing to say. I did a reaction with a bunch of stuff to make more than one product in the same reaction mixture. This is modified, uses modified biodiesel procedure. Those of you that have taken organic chemistry, doesn't look like a lot of you, but it's basically to make biodiesel. You take an oil, you take some alcohol, and use a catalyst, and then it ends up making biodiesel, which can be used as fuel, which is really cool and a very interesting part of green chemistry. And so we took the same procedure, except we used really small oils instead of the huge giant like bacon grease oils that are tons of carbons long. And this way we could limit how long the chains are going to be. And that way we could really analyze what each, what it was going to look like. Got some of my setup here. It changed a little bit over time to make it a bit more efficient, which is always a good thing. And then this down here is the mini GC, which is a piece of equipment that we use to do this. And really, a part of my project was proving that these mini GCs work because they're significantly cheaper than other GCs. Regular GC will cost you probably around $10,000 if it's just gas chromatograph, if not more. I've heard figures that if you add in a mass spectrometer to it, it's around $50,000. This little thing here, all together, around $2,000, a little bit more than that. So it's definitely something that we here can afford, and we have 3 of them now, which is really nice to get people through. But gas chromatograph does a lot of things, but really the main thing that it does is it separates things based on how much they weigh, organic compounds. And so it's helpful because when you see the readout, you can see what each thing is. Okay, this one is just some of my results. I'm not really going to go into explaining how exactly it works. If you want to know, ask me. Jennifer also helped me out with this, doing some of the background work, helping to predict what kind of things will, or what I should be expecting with the products. And yeah, it was an interesting project. People that came up to me actually at the conference really just wanted to know how the GC was. They didn't really care about what I did. They wanted to know how the GC was, but I'm okay with that. And in general, GC was effective.
Okay, on to my project. I did Ester Synthesis. Microscale just means I used really small amounts of reactants and got really small amounts of product, which is difficult when you're washing and you're losing product throughout the reaction. I did it with a heat gun, which can get up to, I think we've found 250 degrees Celsius, which is very, very hot, and you do not want to touch it. So we're using the hot heat gun, as we call it, because we can control the reaction and make it faster. We can control the reaction because we can dictate how long or how hot we get our reactants, whereas using the heating mantle there, we can't necessarily control how hot or how well our product is heated. And also, this is in comparison with microwave ester synthesis, which is also easily done, but again, you can't control how well your reactants are heated. So this is just a little bit of my procedure. We found that imidazole is a really good catalyst for this reaction. And so by adding imidazole and iron hydride together and heating this first, we formed some kind of intermediate that is also helpful in producing the product that we wanted, which is ester. So after we heated the imidazole and the anhydride together and formed this intermediate catalyst, which we have not yet figured out what it is, we added the alcohol, and again, we're able to heat, and we'd get an ester. We also found, though, that we don't have to heat this reaction at all, that just by putting all the ingredients together on the counter and letting it sit for a period of anywhere between 24 and 48 hours or more, depending on what reaction we were doing, we found that this reaction is very green and that it will go by itself if given enough time. So that's the first three there. The procedure also included a wash. The washes help me get any extra reactants or any extra side products out of my ester product. That way I had a pure product to test at the end and to analyze. So this is just a quick analysis using TLC. You see on the left is an acid. On the right is an anhydride, and in the center is my product. Yes, sorry. So that's benzoic acid. This is benzoic anhydride, and this is my product, which is isobutyl benzoate. This is also just a FTIR of my product. The three peaks that really indicated that I had my product is this one at 1700, 1272, and then one at basically 1112. Can't pull off what those mean right now, but they were indicative that I had my product. And so we are on to demos.
So this is some of the stuff at the conference that we went to. Kyle, Dr. Baldwin, and I went to a demo workshop, and they talked with us about making demonstrations more educational and more interactive. And the company that was, our group that was putting it on is called Fusion Science Theater. And they try to make the demo show more of a show than rather just a series of demonstrations or reactions. Their main thing is they have one presenter who talks, and then they have a silent assistant who helps get the audience involved but never talks, which is quite interesting, especially when you see Dr. Baldwin try to mimic it. The one that we saw was the electricity equals moving charges demo, and this is one that they were just starting to work on there. And what was interesting is this race to the glow is related to the paper that Dr. Huntsman and I just got approved for publication. And so at one of my poster presentations, the lady who runs this, who's in the tie-dye here, actually came and talked to us about different ideas for this, which is neat.
Well, they were at the demonstration. Charles and I went to a grad school fair because we were interested in going on to graduate school. I put the universities I have up there because those are the ones that I ended up talking to the most and was the most interested in. So I'm going to tell you a bit about my experience at the grad school and a bit about if you ever get a chance to go to a grad school fair, which if you do, I suggest you take that chance, what you should do. The University of Memphis is the one that I ended up after this. I hadn't really heard of them, but after I went to the grad school fair, I was most interested in, partially because they were most interested in me. I just went and they just had all these tables set up just in this big room upstairs, and all these undergrad students walking around, and there were grad schools at the table. Some of the tables were cool enough to actually have professors there. Those were generally the smaller universities that couldn't rely on just their name to draw people to them, like the University of Memphis and the University of Northern Colorado. The University of Northern Colorado was interesting because their PhD program is not like a PhD in chemistry, it's a PhD in chemistry education with a master's in whatever form of chemistry you're going into, which is really interesting because then it's focusing on not just the chemistry, but on the way to teach the chemistry, which is something that I was actually interested in because I want to be an organic chemistry professor. I want to be able to teach, not just know chemistry. If you go to a grad school fair, or even just go to visit a grad school, there's some things that you should know about yourself and what you're headed to. You need to know what you want to do, like the fact that I know that I want to go on to be a professor, or if you just want to be a researcher, or if you want to go into pharmacology. You need to have an idea of where you're headed so you can ask them some very specific questions about it. Because that was one thing that I found there, was that when I went and asked about if I wanted more teaching assistant experience and slightly research experience, because I want to be a teacher and I want to know that, you know, I can actually teach before I go and teach, just slightly important. There were not actually all that many schools who were interested in the fact that I wanted to learn how to teach and that I wanted to get that experience actually teaching. It was the smaller schools that were interested, so have an idea what you want so you can actually narrow down your field and get out of quite a bit of them. Well, I say where, but I also say be prepared to change. And that's sort of why I have the University of Michigan, because I had mainly universities in Michigan, mainly because those were the universities that I had heard of a lot. But go ahead with an idea where you can visit those ones first, because there's generally a lot of schools there, but that way you can sort of narrow it down and have some time to talk to schools, but also be prepared to visit everywhere else and try to visit everywhere else you want at the time to know why you want to go to grad school and know like how you want to get your experience in, how long you want to go. Make sure, just have tons of questions for the grad school, like what some of their requirements are, what their required GRE score is, what their like required number of years is. The average was 5. But just have some good questions prepared. It's like when you visit a college, but generally more defined.
And while we were there, there was a job fair at the conference, and I'm not planning on going to grad school right away, but I am planning on actually getting a job in the chemical field. And so one thing that I didn't know about the job fair that if I was thinking about it more beforehand, I should have gotten into, you need to start working in advance, because what you can do is you go in and you sign up and you put in your resume and into this system, and then you just apply for jobs and say, like, I want to request an interview with these people who are here during the conference. And I was thinking that was going to be something that I sign up and then later that day they'll contact me because I'm at the conference and offer me an interview. Didn't really work like that. I found out later that you're actually supposed to apply for these things in advance before you even get there. So that way it's just an easy way for them to interview you without having to fly you out somewhere or just do more of an impersonal phone interview. But if you do go, make sure that you have a copy of your resume and cover letters there. When I went, I had a copy of my resume already, but then I had to go back and write a bunch of cover letters because you have to personalize each cover letter for each job. And so I spent kind of an afternoon while they were going to different events just writing cover letters in my hotel room. Not really the way you want to spend this trip to San Diego, so the more work that you can do in advance, the better. So I applied too late. However, this story does have a happy ending in that it's not over. I was contacted 2 weeks ago by Chemical Abstract Service, which they're part of ACS, and I had applied to them while I was there, and 2 weeks ago, that was about a month from the actual thing, I heard nothing from them, and they said that they wanted to set up a phone interview because they had seen my resume on the site and my application. And so I set up a phone interview, and I had that yesterday, and went really well, and they want me back for an in-house interview sometime in the next couple weeks. So, you know, just have hope and cast your line out there as much as you can. There's actually a part on the ACS website where you can just apply to jobs. People post, or companies post jobs to the American Chemical Society page, such that if you are looking for jobs specifically in the chemical field, well, I mean, that's all they have on there. They also probably have some biology type jobs as well, because that's always involved in chemistry, or at least applied chemistry. So yeah, like, take a chance, put yourself out there, put your resume on there. I've also since then joined Monster.com and put my resume on there, and I've actually had people calling me that I didn't even apply to, interested in seeing what I'm like and if I'd be interested in any jobs. So yeah, really, if you want to get a job instead of going to grad school, although you can always go back to grad school, and that's always a good thing to do, and I plan to after I've been working in the field for a little bit. You know, just put yourself out there, and it's interesting to see what comes back.
Sunshine, sunshine, sunshine. Unfortunately, it wasn't always sunny when we were there. Sunday, so we left on Saturday and we came back on Wednesday. Sunday, we had to run through the rain to find dinner and to get to our poster session on time. So it wasn't all sunny, but it was fairly nice most of the time we were there. One of the days we were in San Diego, we actually got to get away from the conference and go visit the San Diego Zoo, which had some really interesting animals there. So it was fun to go see, you know, the pandas and the orangutans and the monkey exhibit, and almost punch a peacock in the face because we thought it was going to attack a baby. So there's this peacock in the pen, but the peacocks just kind of roam around the zoo. They're not required to be in any particular exhibit. And the peacock hops up on a fence right next to a baby. And
it's the top left picture.
Yes.
There.
Right there. Scared probably a dozen people, and a mother with a baby right next to the peacock. It's really amusing to watch. There were other interesting animals to see and watch, and we got to go on kind of just a little cart ride over the zoo and look down on many of the different animals. Yeah, oh, that's what that picture is. Yeah. Okay, right there. We got to watch a monkey and otter exhibit where the animals are combined. Zoos are working towards this, combining different animal groups and combining animals of different species and whatnot in order to save space for the zoo. And it also gets animals to interact with each other in different ways. And so in this picture here, we had a probably 3-foot-long otter and small monkey doing it out, it seemed like. The monkey and the otter were batting at each other, and the monkey would grab the otter and roll it over, and then it would let it go, and the monkey would just kind of watch it, and the otter would go around, and the otter would come up and push the monkey in the water, or try to at least. We spent 10 minutes just watching this epic battle between the two. Anybody else want to take this one? Okay. Also, while we were in San Diego, we got to go to a decent fish restaurant and have fresh caught fish that day, and that was really nice just to get out there. It was a fairly nice restaurant, and we got a seat that could kind of look over part of the San Diego Bay. There were a lot of ships and other things in the water, so. And Pinkberry, I will turn over to Kyle.
Pinkberry is this place that we went to. We got quite addicted to it. It was good that we only went the second to last day, because then we only went twice. But Pinkberry is kind of like Cold Stone, but with frozen yogurt, and all of the toppings are like healthy stuff. And so you well, for the most part, yeah. There's some chocolate and stuff in there, but it makes you feel good about yourself because you're having tasty frozen yogurt, and it's not bad for you. They had peach yogurt that tasted just like biting into like a chilled peach. It was fantastic. And we found that there's one in Ann Arbor, and I think that there's talks about having a Science Advancement Society trip there So, yeah, Pinkberry, it's a big deal. Okay.
Alright, so we decided that it would be nice to kind of tell you how, if you want to go on a trip like this, what you need to do. And basically, you need to live in this building. You have to be really willing to work hard, harder than you think you can ever work, and give more time than you ever thought you had. As I said, mine was a 3-week project. It was supposed to be a 3-week project. I ended up in the dark room for 5 to 6 hours a week, or 5 to 6 hours a day for 3 months, and it ended up in a publication and going to San Diego. I know that Kyle's work is actually his senior thesis he's been working on all year, and Charles ended up spending about 3 months in the lab almost every day for a couple hours. And we put this down here. If you go to organic chemistry, make sure you laugh at Dr. Baldwin's jokes. He likes that. And
sometimes it is a conscious effort, yes.
Yeah.
Another way just to kind of get your name out there and get your face familiar with the professors. The professors here are very into and very interactive with the science club. And so for myself and Jennifer, we both kind of got asked to go on this trip and asked to do research with either Dr. Baldwin or Kyle, because we were involved with the SAS and we'd helped with the periodic table, which had gotten accepted to go. And so we were asked to do research on the side, and so we did research and took that with us to San Diego as well, besides the periodic table.
If you're interested, at the end, we have some things that we have from San Diego, like these wonderful covers that say all of us are the chemists of the year. We each have one. When the light sticks and the periodic table were in the undergraduate research poster, and so they gave us nice little certificates saying, hey, you presented, good job. We also had nice little ribbons that say like poster presenter, chemical education poster session. This one says nano, not really sure why. And we also thought we'd talk a little bit about what our professors did while we were having all of this fun. Dr. Kunzelman actually went as a exhibitor for the microlab systems. I know a lot of you that had Chem 111 and 112 have worked with him in the microlab things. He actually was asked by the founder of Microlab to go and help sell their products at the exhibition. Dr. Baldwin, you might want to ask him sometime about the secret meeting he got pulled into one day.
It was a good secret meeting.
It was a good secret meeting. He was talking with the makers of the Mini GC, and they ended up contacting him and saying, hey, can you come to this meeting? And he got paid to go eat lunch in a fancy hotel. So that was kind of cool. So, do you have any questions? Yeah?
How come the spinach ended up red?
Well, the chlorophyll in the spinach, when it's excited by chemiluminescence, actually fluoresces red. So if you look at the mixture with the lights on, it's green like you expect it to be, and you're seeing the absorbance spectrum. When you turn the lights off, you see the emittance spectrum, and it's red.
Actually, even if you shine the light there.
Any other questions about the science or our random adventures running to Wendy's in the rain? Yeah?
Where was your hotel?
Our hotel was in downtown San Diego,
Coronado Avenue.
6th Street.
6th Street, that's all.
Yeah, it was like 2 blocks from the hotel, so it was nice within walking distance. And apparently we got the only like pet-friendly hotel there, which is kind of crazy because it was like a ridiculously nice hotel, and then you see people walking around with their dogs in there, which just didn't seem quite.
Yeah, if you wanted a bottle of water in the room, it was $6.
Yes.
So, yeah.
How did you know that the Mini GC
Well, what we found was that we could use it to identify the different compounds, because what I did was I ran through standards of the different compounds, and then when I would have the mixture at the end, because with combinatorial synthesis you have like 4 products in the same mixture at the end that you can't really separate. And so you run those through, and they all have, they'll have the same retention times, like how long it takes them to get off of the column that's in the GC. And so since those matched up, like that we could say, oh, this is this, and this is this, and it was very consistent, we know it works. One thing that's different about the mini GC as compared to one of the more expensive GCs is that we can only do fairly small compounds on this GC, whereas I've heard that with the bigger GCs you can run some of those biodiesel oils through that are, you know, hundreds of carbons, or not hundreds of carbons, hundreds of molecular mass units. Yeah, what is that? Yeah, whatever. Yes, Ian. Yeah. You know, it can't do huge compounds, but for the sake of the undergraduate organic lab here, we don't normally use gigantic compounds like that. So while it might not be effective for a industrial chemistry lab, for what we do here, it really works.
And we also discovered that the little handbook that tells you how high it can go is not accurate at all. That was something we discovered. And
we went beyond that.
It went way beyond that. Like it says you can only run up to like 120 weight in the esters. We ran up to like 179, and it was still working, and we could probably run a bit farther than that. It's only, and we actually emailed Bernier about it, and that's only where they got it certified to, safety-wise. They can't go higher.
But if you want to run something through, run it by Dr. Baldwin so you don't ruin the machine.
Yeah.
But
At least without his acknowledgement first.
Yeah.
I think one thing I forgot to mention in my presentation that I was going to, because it's really interesting, esters, you smell esters every day. Perfume, the smell of fruits or the smell of flowers. Esters are made naturally all the time by trees and flowers as they produce fruit or nectar. So when you're smelling something, most likely you're smelling an ester.
And to go off that, with mine, since I also made esters in mine, for you biology people, I did some more application things as well with mine. Check to see if they worked as nail polish remover. Didn't really work that well, unfortunately. But one thing that did work that was really interesting, anybody that's taken genetics in here should know about fly nap, which is kind of an anesthesia for the flies. And it's an amine-based compound, and it'll put them out for like up to 4 hours, depending on the dosage and how many flies are in there. So I ended up using small amounts of my product to see if I could kind of get the same reaction, and it does put them to sleep, which is pretty cool. It puts them to sleep in under 5 minutes, and then they're back up within an hour instead of all of that time. So if you just need to you need conscious flies for a test instead of just examining them unconscious, it might be a good idea. And plus, I found 2 of the products both attract them and then knock them out. So that could possibly have applications for fly traps, in that it brings them in and then knocks them out in a kind of humane way, I guess, if you're terribly concerned about fly civil rights. But yeah, so that was just an interesting application part to it. Are there any other questions?
Well, I guess since there are no other questions at this time, if you see any of us, feel free to ask us questions at that time. We may turn you down since we are science majors and we have many things to do in lab and whatnot. But we can always get back to you if you do have questions. So. And
our posters are upstairs, 3rd floor, down at that end of the building. All 4 of them are up there, so if you want to check them out.
If you have other questions, you want to just crowd around and see the real chemists of the year, come on up. You're out a little early, so hallelujah.